Hello, good morning, students. So, this is about histone codes in continuation with what we discussed earlier. So, histone codes are nothing but it is the modified histones which are going to be read by the different protein molecules. And as you know about genetic codes, similarly, these histone codes are going to be also considered as the epigenetic codes. So, they are going to be plays important role in the earlier, as I told you, conversion of an euchromatin region into an heterochromatin region. So through the process of methylation, these are the few modifications of histones which we have already gone through, like acetylation, monoditri, and as well as methylation, monoditri, and arginine methylation, okay, and serine phosphorylations. So these are the modifications which we are going to see at the histone tiles, so which plays very important role. And these particular modified histones are the one which will get recognized by the chromo or bromodomain having protein molecules, and by binding to these particular side chains they are going to bring about the change in the nucleosomal structure. So either they are going to make them to get very much bundled or very much tightly packed, converting them into an heterochromatin region, or they can make that particular DNA available for the transcription, or they can make the DNA which is ready for getting transcribed into a repressed DNA molecule. So there are hypotheses regarding the modification and how it is actually going to be helpful. So which suggests that distinct modifications of the histones, so it will induces the interactions between chromatin and associated proteins, and modifications may generate independent means it can happen independently, or it can happen means one modification can help in the other modifications in different combinations, and the concentration of combination as well as the modifications are going to plays an important role in the higher order of the chromatin. So there are combinatorial possibilities which are going to be read as a language. For example, acetylation can activate the particular gene, or in other words I can say it is going to activate the transcription, whereas deacetylation is going to cause inactivation or repression of the transcription. Means then I can say we can say that whenever a particular part of the DNA is going to get acetylated, histones are going to get acetylated in the nucleus, means within the chromatin. So we can say that that particular part of the DNA or the gene is being transcribed or is get ready to transcribe. When there is a deacetylation of histones in the nucleosome, that indicates that particular part of the chromatin is going to be get repressed. So means this is going to act as a kind of language to study or in understanding which particular part of the DNA is going to be active in transcription, which particular part of the chromatin complex is going to become inactive for the transcription. So these modifications can act synergistically as well as antagonistically. So one of the modification which we have already seen, which is going to affect the other modifications. For example, here you can see K4 in case of H3K4, when it is methylated, so it is going to block the modifications on the other hand at K9, and even phosphorylation at S10. Okay, acetylation of K14 will happen and DNA becomes active. Okay, so on the other hand, if K9 is going to get converted, then there will be change in the gene expression. So these are going to act together, and sometimes they act opposite also. And whereas we also see that crosstalk between these histone modifications. Okay, the crosstalk is going to happen because the modifying enzyme which is going to be bound are the proteins, the chromo or bromodomain protein molecules which are going to be bound to one particular modified histone tail, can induce a modification on the other tail, as well as it can also remove the modification on the other tail. Okay, for example, acetylation and methylation, they can go hand in hand, whereas methylation, phosphorylation, they are not going to go hand in hand. So like a chromodomain binding protein molecule, for example an R3 in H4, when it is going to get methylated, so by the methyl transferase that can act as a site for binding of chromodomain having hat, then cause acetylation at K5, so which activates the gene. So on the other hand, there is the acetylated uh, lysine can bind with the HMTs, and they can on the HMTs and as well as SNF2s, and which is going to make the DNA become available for the transcription. Like that, the signals are the sorry, not signals, the modifications of histone can talk in between. They can affect each other, okay, and enhance the action or decreases the activity. So this crosstalk between the histone DNA modifications, for example, here you can see there is methyl CAPG, that is cytosine guanine binding protein molecule, so which is going to bind with the methylated residues, and followed by that it recruits the HDAC, so which deacetylates the histones and makes the histones makes the DNA not available for transcription. On the other hand, also recruits the HMTs. Whereas we see the same MBDs, they can also bind with the SET protein molecules, which are going to cause methylation, and or there is one more like chromobindomine protein molecule can bind with the methylated residues, and further they can cause DNMTs, so which is going to methylate the other bases. Means this crosstalk can cause further modification of histones and the DNA. The reading and translating this particular histone code is a very important process, which is done by both bromo and chromodomain protein binding protein molecules. There are about 75 chromodomain chromodomain proteins, sorry, bromodomain proteins in humans, and chromodomain proteins, they recognize these modified histones also, and they are going to help in the process of understanding the means helps the DNA to get transcribed, or stops the transcription also. So the writing and reading of these particular histone codes, authors of histone codes are HMTs, kinases, HATs, PPTases, that is phosphotransphorases, HDACs, HDMs. HDM is a question mark. The methylation demethylases are not being found much. Whereas these are the authors, means they are going to cause a modifications. There are readers, that is chromo and bromodomain proteins are called as readers, which is going to read these particular modified histone tails as well as the bases, and they are going to behave in that particular, means in a specific manner. So based on the kind of modification, based on the kind of chromodomains or bromodomains. So these are all modifications of histones which you can see together, which is going to convert having very large biological implications in terms of activity of the DNA. So there is one small thing like histone deacetylation. People have studied this histone deacetylation and the HDAC activity. They've used these repressors called MADMAX. So MAX is a kind of protein molecule which is going to act as activator as well as inhibitor based on the uh, other part, which is going to be bound with the MAX. So when MAX is bound with the MAD, so it is going to act as a repressor. So there are co-repressors like N NCOR smart combinations, that is nuclear receptor co-repressor or silencing mediator of retinoic acid and thyroid hormone receptors. So these are going to be involved in activating the DNA, which is bound with RXR, that is having RXR, that is retinoic acid regions, sorry, binding regions within the DNA molecule, and they bind with the retinoic acid binding, means that particular dimeric proteins, they causes the repressions. So there is another protein called as SYN3 which is also going to be involved in the activity. So all these repressors, co-repressors, what they do is they call upon the HDACs, so causes deacetylation. So when HDAC is going to deacetylate, so automatically the DNA is going to become unavailable for the uh, transcription, thereby they repress. You can see here how they are going to interact with RXR protein, sorry, RAR, retinoic acid receptor and retinoic acid protein activated proteins. They bind with the DNA, so having specific regions to the enhancer region, then they call upon INCOR and SMART together, they call upon HDAC, and which causes deacetylation of the histone side chains, and repression is going to happen. So people have studied this modification, repressor activity, and both activation and inactivation of the DNA. What they have done is they have taken thyroid hormone response element regions, and they have understood that. So this thyroid hormone response element regions, if they are going to be covered with a nucleosome structures, for example, when the thyroid hormone, the presence and absence of the thyroid hormones is going to play a very important role in the activation or inactivation of the particular this thyroid hormone response genes. So for example, whenever a thyroid means this TRE, when um, it can act as a site for binding with the retinoic acid receptor is protein is going to be dimerized with the thyroid hormone receptor regions. So this protein is going to be usually bind with the TRE regions. So once bind with the TRE, in the absence of thyroid hormone, they can cause repression of that particular part of the DNA, where they are going to call upon the histone deacetylases through NCOR and SYN3H combination. They call upon HDAC, which is going to cause the deacetylation of histones, thereby it is going to induce the repression. On the other hand, there is in case if the thyroid hormone is there, so that can go and bind with its receptor, the thyroid hormone receptor, which is dimerized with the retinoic acid receptor protein, which together bind with the TREs. So now when the thyroid hormone is there, so that instead of recruiting NCORs, synthetic dimerization dimers, it will now call upon transcriptional activators or co-activator proteins like CBP300 or PCAF protein molecules, which together calls upon an acetylation protein called T5 TAF2 250, so which is a histone acetylase, and which further acetylates the histone side chains. And you can see there, when it is inhibited, deacetylation causes all these histone tails to tightly bind with the DNA. On the other hand, when they get acetylated, they loosely bind and they free that particular DNA now, which is available for binding with the transcriptional proteins, which can induce the initiation of transcription. So that's a way in which the histone deacetylase is going to be involved, and the modification is going to happen. So based on the availability of a different proteins, like here hormone is going to act as important signal for the activation or repression of the genes associated with the hormonal activity. So this is how the modifications are all going to, this is how the histone modifications are going to take place, and that in turn is going to help in the activation of transcription. But when you say histone modification and activation of the transcription, we cannot exactly means straight away say that the histone modification itself will be sufficient enough to drive the transcription. See, histone modification is going to be considered to be a first step during the process of transcription, either activation or repression. Okay, so when you say activation, so earlier if you remember, I showed the experimental components as well as the pictorial representation, wherein you have seen the nucleosomes sometimes they are going to be having completely covered with the promoter region of the genes. So when they are completely covered the promoter regions of the gene, due to the modification of histones, these promoter regions may get available for the binding of transcriptional initiators. But in order to elongate and as well as to complete the transcription, you need the whole part of the gene to be free, so that easily polymerase can able to produce the RNAs. So for that we need these particular nucleosomes to reposition, or in other words to change its position, either to get away from the DNA molecule, set the DNA free so that the transcriptional proteins can involve in the process of transcription, or even I can say replicational process can easily takes place. But uh, when but for the or else it can also do that, it can actually get completely removed. The nucleosomal proteins can undergo means disassemble, and then thereby they can also set the DNA free. So for this particular process is nothing but it is a remodulation of the DNA, or remodeling of that particular chromatin structure. So for the remodeling, that is either to move down the nucleosome or move up the nucleosome, or in other words also for to cause the disassembly and assembly of the nucleosomes. So it requires the proteins which are going to be able to change the chromatin organization. So this change in the chromatin organization is the remodeling of the chromatin, so which is a very very important and first level of regulation of gene expression, I can say, in case of eukaryotes. So there are several protein complexes which are going to cause this remodulation of chromatin, and all are going to use ATP, means energy from ATP hydrolysis, and remodeling complexes are distinguished by their ATPase component. So the remodeling mechanism, if you want to, means when we want to study, sorry, one second. Okay, so sorry, it got little stuck. So you can see here in the mechanism of chromatin remodeling. So it can happen by either sliding, wherein you can see that orange region is nothing but it is the promoter region covered with the blue nucleosomal structure. So the remodeling can involve in the sliding, where just like you can say, so you can draw the thread which is tied around a particular means a static structure. So when you draw the pull the thread will come out of means it will roll around the particular component and comes out. So like that sliding of that thread, that is here the DNA can happen around the nucleosome, or even ejection. So in the ejection that whole nucleosome can get eject, so giving leaving the DNA free, so available for the process. Or even there can be another way, instead of complete nucleosomal ejection there can be even the H2A H2B dimer rejection, so which is also going to cause the nucleosome to get disassembled partially, and set the DNA DNA becomes more accessible for the transcription. There's when for example require the DNA is completely accessible, process is completed, again an H2A H2B variant can also come in there, and they can interact with the DNA, and their variants can also have a different kind of affinity for the DNA, which also makes it accessible, and also it can help in the remodeling of the chromatin. So if you come to the nucleosomal sliding, I don't know what is happening. So I think this is sorry, it is may not be supported well. So you can see when you see the, I'll send you this particular slides also, you can see. So what exactly is going to happen here, you can see their the ATP is going to be very important for the remodelation. People have studied by providing ATP in the absence and presence of ATP how the remodeling is going to get affected. So when a remodeling complex is added to a sufficiently modified region of the chromatin, so histone modified region of the chromatin, the remodelers usually actually requires the histone modification, which is because these remodelers are going to be either chromo or bromo, chromo or bromo domain having proteins, or these proteins can interact with chromo or bromo domain proteins, and thereby they are going to sit on that particular part of the chromatin structure, and then they can cause remodelation where they can change the relative position of the nucleosome, few nucleosomes, and or else they can decrease the means eject the nucleosome and make the DNA more accessible. So nucleosome conformational change sliding provides efficient way of following the DNA to be accessed, and generally people have found that this is done by one type of chromatin modeling protein called as SWSNF complex, that is called switch sniff complex, which later I'll discuss further. And it creates a means it frees the DNA molecule, means set the DNA free. The average loop size on the nucleosomal template is approximately 100 base pairs. The second chance is nucleosomal ejection. So here ejection is going to set the DNA free. So DNA loops and removes the dimer, or even entire octamer can get removed. So the tendency of octamer to dissociate into H2H2B dimer and then H3H4 tetramer, it is there, and large loops may allow the DNA, other DNA molecules to invade and open the histone DNA contacts. That can also happen, and DNA translocation releases the neighboring histones dimer or octamer can also takes place. Then other ways like remove the histone from the DNA, so you can see how it is going to set the DNA free by the histone eviction. So you can see how the process will take place, proposed mechanism. So that's a nucleosomal structure. The Gal4VP16 protein binds to the promoter region, calls upon modifiers, or and then modifiers calls upon the remodelers. You can see ATP dependent remodelers bind and causes change within the histones, makes the dimer H2H2B to come out, then tetrameric is going to come out. The complete nucleosome is goes off, leaving the DNA, the twinned DNA molecule, intertwined super coiled DNA becomes free. So histone exchange catalyzed by SWRI complex catalyzes the exchange of nucleosomal H2H2, means H2A for H2A Z variant. So release of H2A ZH2B from the SWRI complex causes the relaxation of DNA, and it also causes reassemble of histone octamer. You can see replacement of the normal histone protein for the variant, which sets the DNA a little more free. So histone tiles are going to be very important because it will have it will act as a site for binding with the remodeler complexes. Modification means makes most it recruits the remodeler complexes and stability is given. Histone acetylation stabilizes SWSNF interactions with the nucleosomes. These are the remodulated histones. You can see how exactly that will remodulation happens, how it is going to affect the DNA availability for the transcription, and the package also you can see the tightly packed earlier core stones, and when the remodulation happens they become more loosely bound. This part we have seen already. The stellated histones allows the transcription to begin, and you can see here the reactions catalyzed by the ATP dependent chromatin remodelers, how that will affect the process, can cause the DNA to be available for transcription by the remodeler activity, which can happen by ejection or repositioning or unwrapping and even dimer exchange or dimer ejection. So this is again how remodelers are working. So dynamic range of chromatin structure created by ATP dependent chromatin remodeling. So it causes changes in the dynamic changes, means the structure of the chromatin is going to get a little bit altered at a particular region. So distance between the nucleosomes will increase, and somewhere distance between the nucleosomes we can decrease. So many things happens. So we'll stop here. We'll see about the types of remodelers and how remodelers are going to work in the next coming class, next videos.